Hello everyone, I am Ashish and this is my channel Medical Acme India, and today we are going to discuss sixth lecture of our lecture series which is going on, that is Animal Diversity lecture series. And we have assembled here to discuss Sarcodina, in which we will discuss mainly about the amoeba and Entamoeba. You all know Sarcodina is what? A superclass of kingdom Protista. So guys, what happens? Sarcodina is divided into two classes, and it is like Rhizopoda and the another one is Actinopoda. So Rhizopoda may we will consider amoeba and Entamoeba. So these organisms are very important pathologically, and also in our curriculum it is defined in many syllabus. So we are going to discuss these two organisms here. So let's start. Guys, first of all, amoeba is very common, and it is you all know is found in fresh water, and it is fresh living. It means that it is found in fresh water, means fresh water, pond and river you will find it. And in free living, what does this free living mean? Free living means it is not dependent on any kind of organism for like parasitic in nature or any kind of like hostile or anything. And the second important thing is here to note that it was discovered in 1755. It is very important to note that in 18th century, like in early 18th century, it was found, and it was discovered by Rosenhoff. So guys, it is artificially grown, or you can say cultured by the method of hay fusion method. What is this hay fusion method? Hay fusion method is a method in which what you do, you just collect pond water or river water, that is fresh water from any source, and then keep culturing them in the lab. So what does this mean? This means that E. coli can be easily grown, and it is very much of interest in research. So guys, what does taxis mean? Taxis is the property of amoeba. It is the worst organism in which the taxis is seen. Taxis is something in which it directional. Kinesis is non-directional, but taxis is directional, as you all know. Directional movement is seen toward the stimulus. Stimulus can be any environmental stimulus, galvanic stimulus, or anything. And the main important thing is amoeba has digestive enzymes like those found in ours, like most of the vertebrates. They have the digestive enzymes. These digestive enzymes are very important for the like engulfing, or you can say when they eat up the food, or you can say phagocytose the food items. Then what they do, these digestive enzymes comes into play, and they like break down these molecules. So is the assimilation and absorption of the energy occurs. So guys, the cytoplasm of the amoeba is divided into two main layers, that is ectodermal layer and endodermal layer. Endodermal layer is again divided into two like compartments, that is first one is the gel and the second one is the soul, that we will discuss in the later, like when we will discuss the movement in amoeba. So guys, pseudopodium, as you all know, Sarcodina is the family where locomotion occurs through the pseudopodium. So pseudopodium can be of two types. First one is the lobopodium, and second one is the monopodium. Lobopodium means lobes, many lobes, or you can say the polypodia is seen. Polypodia means multiple pseudopodium is seen in the organism. It is not the case, as you will see in the next slide, that Entamoeba has the monopodium, that is it has a single pseudopodia from which the locomotion occurs. Plasma lemma is the outer boundary of the cell. In all, you can say amoeba and Entamoeba, all the Rhizopoda has the like pseudopodium as the outer layer, or it is the covering. It is what? It is a thin elastic membrane. It is also the lipid bilayer, but it is very thin in nature as compared to the plasma membrane. So abundant food vacuoles are found in the organisms, and contractile vacuole is the property of freshwater organism, where what they have to do, they have to maintain the osmolarity. So contractile vacuoles, it is very important for the NEET guys and CSI and ICMR guys, where it is directly asked that what is the role of contractile vacuole in amoeba, or you can say in the freshwater organism. So contractile vacuole has the role in regulation, where it maintains the like saline and buffer all. So guys, Amoeba Proteus is very common, and it is very widely found in the fresh water. So it is the structure of these organisms are given. Here you can find that all of the like basic organelles like nucleus, plasma membrane, that is plasma lemma, and all the mitochondria, everything is found. But what here to note, that here you have to note that the food vacuoles are present in abundance and contractile vacuoles are present. So these are the two like things, and everything which is like ectoderm and endoderm, ectoplasm or endoplasm is discussed previously. So guys, let's move toward the soul gel theory. What does soul gel theory says? Soul gel theory was proposed by Hyman first, and it was supported by the Pantine and Mast. See what happens when you are doing in a research and you are indulged in research, then what happens, the same research is going parallel in many universities or many labs. So what happens, they independently discovers the things. Suppose by chance they discovers the things in the set at the same timeline, then they both, the scientists who have discovered the things in the same timeline, they are given the credit. So supported by Pantine and Mast independently. So independently, here is the very important thing which you have to note. So it is for the locomotion of amoeba. This theory was in concern with the locomotion of amoeba. Locomotion means movement in amoeba, how amoeba moves from one place to another place. So it is a physico-chemical process. What does this mean? This means that the physical and the chemical things all are involved in the organism. Chemical things may protein, biomolecules, everything is covered. And physico means the physical part, that is the you can say experimental part where the evidence is seen. So what happens, contraction and regulation of proteins, many proteins are here, like they are basically the cytoskeleton system which are seen in the organism, and that the contraction and relaxation of that protein gives rise to the movement of the organism. Solation and gelation, this is very important to note here, that thus amoeba has, as you will see here, that the amoeba has cell and gel and soul. You can see here that the amoeba has two layers in the endoplasm. First one is the gel and second one is the soul. So guys, let's discuss it very like you can say in descriptive way, where you are seeing that the first is the uroid. Uroid is the posterior process of the pseudopodium, and there are two ends, as you can see, trailing end and advancing end. Advancing end means the direction in which the movement is going on. So the zone of gelation and zone of solation become important. What is zone of solation? Zone of solation means that gel is converted into soul, and gelation means soul is converted into gel. Soul and gel differs in the viscosity. It has like a kind of thing in which, suppose if you are given a slurry kind of thing and water, then what happens, slurry will like move very slowly and water will move very fast. So this thing here happens. So what happens here, you see, first, as you will see at the leftmost arrow, you can see that the gel is there. Then what happens, the soul part is at the anterior or advancing end, you can see. So gel pushes the soul, and then what happens, the direction in the advancing end occurs. Gel has the larger, you can say, density. So what happens, they pushes the soul or the liquid or fluid part of this. Then what happens, the soul, as you can see many arrows in between, like in the center of the cell, center of the pseudopodia, they just go and just have the bidirectional movement of the soul around the pseudopodium end, or you can say the apex of the pseudopodium and bottom of the pseudopodium. As you can see at the anterior end, that two arrows are given. These two arrows denote the bidirectional nature of the solation, or you can say the soul movement. So this is very basic about the soul gel theory, and you can remember it. It is very important. Here the only thing which is asked is the who discovered it, like Hyman, and what is solation and what is gelation, and it is also it is a physical process, physico-chemical process, or anything. So this can be remembered. This is very important. So guys, let's discuss about our main focus organism, that is the Entamoeba histolytica. Entamoeba histolytica is the organism that is belonging from, as you all know, Protista, and phylum Protozoa. Superclass is Sarcodina, class is Rhizopoda, and genus Entamoeba, and species Histolytica. These are the main highlights which you have to remember. You can remember it. So guys, it was discovered by Lambley, and the pathogenesis of the disease was described by Lost. So Lambley is very important to here remember for NEET guys. Many times it is asked. It causes actually amoebic dysentery or Amoebiasis. Amoebic dysentery means dysenteric actually. This means dysfunctioning, and enteric means intestine. So intestinal dysfunctioning is seen in amoebiasis. So guys, amoebiasis is very important. It is very common in most of the, like you can say, in common infection, you can say. It is the amoebiasis. Amoebiasis is seen many times in many organisms, in vertebrates and vertebrates all. So guys, the main thing here to note that it is the monogenetic parasite. It means that it is not transmitted from any animals. It has the same host where it completes its all life cycle, and that is human. So guys, human it completes its full life cycle and infect human. So guys, it is inhabitant means it is found in the human colon area of the large intestine of vertebrates, and invertebrates maybe these are found. Pseudopodia is again monopodium, as I have said in the previous slide. Two forms are found here, minuta and magna. Minuta are immature form, or you can say the premier, or you can say the organism which just came out of the binary fission. Magna is the adult form, and it is also called trophozoite. Trophozoite is pathogenic stage. Quadrinucleate stage, as you will see in the next slide, that it is the infective stage. What does this infective stage mean? Infective stage means when you are just here and what the infection comes, like suppose the quadrinucleate stage is the stage which is actually transmitted from the oral, or you can say the contaminated food, through the in our body. So it enters as quadrinucleate stage from the food items, or contaminated food items basically. So guys, you can see here that the structure is given, minuta and trophozoite. In minuta you can see that the plasma lemma is there, endoplasm is there, ectoplasm will be there, and nucleus is here. And the main important thing, it has the chromatin granules. Chromatin granules are, you will see in the next slide, it is very important for the infection point of view. So guys, another is the trophozoite or magna form. In magna form there are highlights which is like pseudopodium. You can see here that the monopodium is very highlighted, and endoplasm, ectoplasm here will be found. And the most important thing here is the food vacuoles with bacterium and RBC. So RBC, which is engulfed, or you can say in course of what happens in course of like infection, it shows their nature, and the nature of the trophozoite is that it has the phagocytic nature. So phagocytosis is something, as you all know, is the eating of anything from outside the cell or environment, you can say. So what they do, they engulfs RBC. So guys, RBC enters the cell and they are broken down. So what happens here, the anemia kind of thing is seen. So they actually target the RBC in the mucosa and submucosa region of the intestinal anatomy. You will say it has the mucosa, submucosa, and circular muscle, all the things, serosa, all are found, but it infects the mucosa and submucosa region of the intestine. So guys, let's discuss about the life cycle. It is very easily drawn here. So guys, you can see that you can start from the binary fission, that is rightmost here. What they do, the trophozoites are like and undergoing the binary fission, and then what happens, the pre-cystic form or minuta form. Minuta is a smaller form, so it has the minuta there. So they are smaller forms, and the main feature here is the chromatoid body. What is chromatoid body? Chromatoid body are the aggregate or accumulation of ribosomes. They are actually the clumps of ribosome, you can say in layman language. So what happens then? Then the minuta undergoes cystation, or you can say encystment. And encystment is nothing but the formation of covering outside the organism to protect from environment. So this is the indication that it is going to be in an important process, or going through a, like you can say, a life form. So here that life forms starts with the karyokinesis. Karyokinesis is marked by the binucleate cyst and then the quadrinucleate cyst. This quadrinucleate cyst, I was saying, is the infective stage, or you can say it is transmitted from the food items, actually the contaminated food items, from one person to another person. So guys, you can see here the 4 nucleus are there, and the abundant glycogen vacuoles are here, which will be required for the karyokinesis. So guys, then what happens? Then another step comes here, excitement occurs. Excitement is nothing but the you can say the exo means coming out. So coming out from the cystic wall occurs of the organism, that is here the trophozoite. So sorry, minuta. Then the minuta comes out, and it is now called the metacystic amoeba. Metacystic means now it is prepared to undergo the division. So guys, the another division occurs, and there are 8 actually trophozoites which are formed from a single minuta. So guys, this can be remembered that 8 trophozoites are formed from a single minuta, and these 8 trophozoites are called metacystic trophozoites, and they then undergo the maturation process, and then differentiation occurs. And then what happens, then the adult trophozoites comes into existence, as you can see here. The adult trophozoite is something which has the nature of phagocytosis, as I said. So they engulf the RBC, and then the infection starts in the mucosa and submucosa layer of the intestine. So here it is very importantly noted that the mucosa and submucosa mucosal RBCs are actually targeted in the organism infection. So guys, this was all about the life cycle. Let's move toward some of the another species which are non-pathogenic, that is Entamoeba coli. It just enhance or modulates, you can say, the digestive process or absorption process, and it is the resident of gut without any harm. Entamoeba gingivalis causes intestinal abscesses, or you can say intestinal ulcers. So gingivalis is also infectious, but some of the non-pathogenic species are also found in the Entamoeba. This is to remember, and Entamoeba coli is one of them. So guys, these are important questions from this topic. You can attempt it and enjoy it. It is very important. They are listed in very like you can say compact way, and just solve it and enjoy it. So guys, thanks for loving my channel, and please subscribe, like, and most importantly share the channel to the needy ones. If any one of the individual gets benefited, then I will be very much blessed like thing. Thank you so much guys.